Hello students, welcome to my biology class. This session is for students of class 12. Today's discussion is on chapter 5, Principles of Inheritance and Variation. Today is the first day of this chapter, so we will be having a discussion on Mendelism. So today's topic is Introduction to Genetics, and the specific area will be on Mendelism. So let us start the discussion. At the beginning, let us know what you will be learning, and at the end of the session, what you will be able to do. That means these are actually your learning objectives. So at the end of the session, you will be able to define several terms like genetics, inheritance, variation, and many more. You will be able to apply the different terms related to inheritance. You will know and will be able to write why Mendel chose garden P for his experiments. You'll be able to analyze the results of monohybrid crosses and other crosses too. You'll be able to apply the concepts of segregation in any cross. You'll be able to prove that the law of segregation is universal, and you will be able to explain the law of independent assortment. Now let us come to some important initial terms before we enter into the details. Now, since this is a topic, this subject is genetics. So several such terms like genetics, variation, inheritance, heredity, we must know about this. What are these? In very brief, I will discuss. Genetics is the scientific study of mechanism of inheritance and variation. This term was coined by Bateson in 1906. Heredity is the transmission of characters, resemblances, and variation from one generation to the next generation through genetic descendants. So here it is important that it is through genetic descendants. Next term is inheritance. What is inheritance? It is the process by which the offspring acquires the character of its parents or the cell. If it is a cell division, then of course the cell, otherwise the offspring, they acquire the characters from the parents. But during this process of inheritance or acquiring the characters from the parent, certain amount of variations may occur. Then what are these variations? These variations are actually some deviations from the standard form, which can be between the cells, means the parent cell and the daughter cell, or it can be between the parents and the offsprings in a particular species. So these certain differences or deviation may be caused during inheritance, or it may be due to some environmental factors. So here we just have a very brief discussion on the four terms. Let us first know who Mendel was. Mendel was an Austrian and was born in a poor family. His father was a peasant in Moravia. He became a monk at a very early age. So he was born in 1822, and in 1843 he became a monk. In 1847 he became the abbot. You know what is abbot? It is the head of the monastery. So abbot means the head. So in 1847, Mendel became the head of the Augustinian monastery of St. Thomas in Austria. Now it is in Czech Republic, Brno. In 1851, he went to Vienna University and he studied natural history and mathematics for 2 years. After that, he returned in 1853 and started working as a teacher of physics and natural history. During this time of 1856 to 1865, he conducted the breeding experiments on pea plants. In 1866, he published his findings in the annual proceedings of Natural History Society of Brunn. However, his work was not recognized during his lifetime. After his death, it was recognized. We'll discuss more in details later on. However, he is still today remembered as father of genetics. Whatever advancement we have done as on date, the basic lies on Mendel's work. So he is remembered as father of genetics. Now the question is, why Mendel's work was not recognized during his time? Actually, certain problems were found. That is, he published his work in a journal which had limited circulation. Just now I told that he published his work, but the journal had limited circulation, due to which most of the people were not aware of his work. Secondly, his mathematical approach in solving biological problems was not convincing to the contemporary scientists. Mendel was quite ahead of his time, actually. Since he studied maths and natural history, so he could apply the statistical knowledge in solving biological problems. But this was far ahead from the contemporary scientists, and so they were not able to understand this, and they felt what is the role of biology in biology, this mathematical application or statistics. So they couldn't relate this. Next, the minds of the scientists at that time was actually preoccupied by Darwinian theory of evolution. Charles Darwin's theory of evolution at that time was in the top ranking information, and so people were highly preoccupied by this theory. Whereas Mendel's idea of heredity and variation was quite opposing the Darwin's concept. So this was not easily accepted or could not be accepted. He could not prove any presence of the factors. Actually, Mendel considered that factors are responsible for the traits and inheritance. But when he was asked, or he could not prove that what are these factors, where it exists, how it exists, what is their composition, so that part he could not explain. His concept of factors as discrete units could not be accepted since variations still occur. Mendel did not believe in blending, and so there was a confusion because there are such variations in nature which Mendel did not explain. Furthermore, his results were not successful on other plants. Why his results were not successful on other plants? One of the reasons may be Mendel worked on pure lines, whereas others might not have worked on pure lines, or they might have worked on hybrid plants. So Mendel's law could not be applied on those experiments. So these were some of the basic reasons why Mendel's work was not recognized during his lifetime. There are more points, but these points which we have discussed is sufficient for this moment. Before Mendel, there were many concepts. We don't have such scope to discuss all of them. Just I will discuss one or two. Number one is blending inheritance. What is this blending inheritance? What happens is the qualities of the parents are mixed in the offsprings. That means in blending experiment, the father and the mother, their characters passes to the offspring. The offspring now will bear the mixture of the two characters. For example, if one of the parent is dark skinned and another parent is white skinned, then the child or the offspring is supposed to have intermediate between the dark skinned and the white skinned. This is blending inheritance. Next comes the theory of pangenesis. This is theory proposed by Charles Darwin. He proposed the idea and suggested that Every part of the body releases some microscopic entities. These are called as gamules or pangenes. These gamules or pangenes are transmitted through the gametes into the zygote. It is these gamules or pangenes which guides the growth of the relevant organs in the body. Next is continuity of germplasm theory. August Weismann suggested that living organism consists of germplasm and somatoplasm. Somatoplasm means the part which makes the whole body, and the germplasm, these are transmitted from generation to generation, and it is the germplasm which maintains the continuity of the race. So these were some of the experiments or concepts before Mendel's. This is not the list. There is a long list. There are many more concepts which we are not going to discuss right now. Next issue is, why did Mendel choose garden P? There are several reasons for his selection. We'll discuss some. You can see here the garden pea. All of us have seen this, and this is a very common plant. So they have easily detectable traits, easily identifiable, and very prominent. Each character have contrasting traits, and such contrasting traits are very prominent. Flowers are large, bisexual, and self-pollinating. Easy to hybridize. These plants have short life cycle, easy to cultivate, and also to preserve the seeds. These preservation of seeds are required because each seed is one generation. The seed when sown in the next season actually gives the result of the previous activities. Let's now discuss monohybrid cross. What did Mendel do? Let us first try to understand this. Mendel was the first to carry out experiments in planned way and also conduct quantitative study of inheritance. He used Pisum sativum, a garden pea, for his experiment, and he published his findings in 1866. Mendel studied several characters and traits of pea, but finally he selected only 7 characters to formulate the laws of inheritance. Here one thing is very important which you must remember, that is, he selected only the true breeding varieties for his experimenting. He conducted controlled self and cross-pollinated pollinations, or otherwise called as hybridization, and maintained the records of every cross and generations. These are some of the important activities which he has done. Now, in the previous discussion, we just found cross-pollination and hybridization process. Let us first understand what is this hybridization process. Cross-pollination or hybridization. Cross-pollination means, that is, transfer of pollen grain from anther from one flower to the stigma of another flower. Since in this figure you can find, since this flower, pea plant or pea flower, this is a bisexual flower. It has petal, it has, there is the stigma, anther, stamen. The stamen has anther, and there is the carpel, there is a stigma, style, and ovary. So this is a bisexual flower. In a bisexual flower, there is every possibility that there will be self-pollination. So to prevent this, some control mechanisms have to be taken. To do any hybridization process, the first step is selecting the plants for hybridization. Plant A, plant B of the same species, so they have to be identified. This identification includes transferring pollen grains from which plant to which plant. Accordingly, the second step will be emasculation. What is emasculation? Here you can see, that is, with the scissors, they are cutting off the male part, that is the stamens. So emasculation means early removal of anthers. The anther removal is required so that at any cost there is no self-pollination in the hybridization process. So this will prevent self-pollination. Next is, as we have already selected the plants, so dusting of pollen grains. So I know which is the plant A and plant B. I want to transfer the pollen grain from plant A to plant B. So I will collect pollen grains from plant A and dust the pollen grains on the stigma of plant B. So this is dusting pollen grain over the stigma of the predetermined flower. After that, the flower has to be covered with a bag. The bag may be made of butter paper or some other material. This is required to prevent any unwanted cross pollination because we are conducting cross pollination or hybridization under a controlled process. Now let's see what were the stages of experimentation. What did Mendel do in sequence? So in stage 1, that is selection of true breeding varieties. How these true breeding varieties can be obtained? That can be obtained by successful or self-pollination, successive self-pollination, and finally selecting the true breeding offspring. So ultimately, he could select 14 such varieties. These 14 such varieties are actually 7 pairs of contrasting characters. So he selected 7 characters, and for each 7 character, for the 7 characters, each character having 2 alternate traits. So that means 14 traits of varieties were selected. Next, he conducted reciprocal crosses between the pure plants. I told you earlier that Mendel's experiment is based on purity. That means pure plants. For the character he had chosen, the gametes must be pure. That is, when it is a pure plant, reciprocal process, here he got the same results. What is a reciprocal cross? Reciprocal cross means A cross with B, a plant A cross with plant B. That means pollen is transferred from plant A to plant B. Then certain results will be obtained. If the same is reversed, pollen is transferred from plant B to plant A. Certain results will be obtained. In such cases, these are called as reciprocal crosses. So A to B or B to A, such experiments are or crosses will be called reciprocal crosses. In Mendel's experiments, reciprocal crosses gave the same results. So after every reciprocal cross, he obtained the seeds. Because after the cross is made, pollination is over, then fertilization occurs, then fruit and seed develops. Seed is the product of the cross. Now, what character has been inherited in the seed has to be checked, and this is possible when the seeds are sown in the next season and then the plant grows and then the characters may be observed. So these seeds, which is the result of the crossing between the two parents, is the first filial generation or known as F1. So after this, what he did, he grew the F1 hybrid seeds and obtained, and he could find out the result of the cross in the previous session. A previous season, whatever he had done, the result comes in the next season when the seeds are grown. Now, in the next step, what he did is he made crosses between the F1 hybrids. That is self-pollination. So when F1 hybrid is crossed with an F1 hybrid, then again some results are obtained, and those result is in the F2 generation or the second filial generation. That means again the seeds have to be harvested, they have to be preserved and grown in the next season. In the next season, as the seed grows, the phenotypes can be observed. So Likewise, the same can be continued to the third generation, and he did so and analyzed the results. Traits and characters Mendel chose for his experiments. What is a character and what is a trait? Let us first understand this. Character is the heritable feature, and each variant of the character is a trait. For example, height of the plant. If I say height of the plant, it doesn't make any sense until I say it is either tall or dwarf. So these are the contrasting traits of this character, height of the plant. Similarly, position of the flower is a character which may have axial trait, whose position of the flower may be axial, or position of the flower may be terminal. So the trait expresses the appearance. This trait may be a dominant trait or can be a recessive trait. Here are the 7 such characters Mendel had chosen for the experiments. Height of the plant, these are the dominant traits, and these are the recessive traits against their respective characters. Like height of the plant, tall is a dominant trait, recessive is dwarf. Similarly, shape of the seed, if it is round, it is a dominant trait. If it is wrinkled, it is a recessive trait. So you can see here in this picture also, these characters are shown as dominant traits and recessive traits. So this is a very important question which is very frequently asked. What are the 7 characters Mendel had chosen for his experiment? And write in a tabular form the dominant and the recessive traits. Here you must not confuse in this position. That is, in certain cases, there may be some confusion. For example, color of the pod. If it is green, it is dominant. Whereas color of the seed, if it is green, it is recessive. So, color green in the pod or in the seed, their dominance and recessive traits are different. Green pod is dominant, whereas green seed is recessive. Otherwise, the remaining characters are easy to remember. There is no confusion on this. Let us find Mendel's observation. What did he find? He found, for example, let us think of a tall plant being crossed with a dwarf plant. Then what he should have obtained? He found that the F1 hybrids were similar to either of the parents. When I say tall plant and dwarf plant, since we have already understood, as we have studied in the previous slide, the tall plant is dominant over dwarf plant. But how did he obtain this? When he crossed a pure tall with a pure dwarf, he found that the F1 hybrids were either similar to the parents. That means the F1 hybrid will be either tall or either dwarf. Whatever is expressed in the F1, he considered that as to be a dominant trait. And the one which is hidden is recessive. In this case, if the F1 would have been dwarf, then dwarf would have been a dominant trait. But it was not so. What he found? He found 100% tall plants. So he considered tall as a dominant trait. Similarly, for all the 7 characters, he conducted experiments and finally he could come to a conclusion for each character what is the dominant trait and what is the recessive trait. While doing such experiment, he performed reciprocal crosses and he found that reciprocal crosses gave the same results. Again, after getting the F1 hybrids, he conducted the next step, that is, he crossed F1 with F1. He found that the F2 phenotypic ratio was 3:1, which means 75% of the offspring were dominant type and 25% were recessive type. Now, to his astonishment, he found that the recessive trait, which was hidden in the F1 generation, expressed in the F2. Now, again, let us go back. In the parental combinations, where tall plants were crossed with dwarf plants, there was no dwarf plant in the F1. So F1 plants were all tall. Now, when tall plant was crossed with tall plants, what should be the result? Here, he found 75% were tall and 25% were dwarf. Now, when we counted the number of plants, he found that in the F2 generation, he got 787 tall plants and 277 dwarf plants. From this, he came to the ratio of 3:1 ratio, phenotypic ratio in the F2 generation. Now, while analyzing the F2 generation, again, he found that 3 out of 4 were tall and 1 out of 4 were dwarf. But the 3 tall plants, they were not pure tall plants. He realized that only 1/4, means out of 3, 1 was pure tall and 2 were hybrid type. So now he found that the 3:1 phenotypic ratio is actually having different genotypes. 1/4 are true tall, 2 out of 4 are tall but was hybrid type, and 1/4 of the population was true breeding dwarf type. Now he could get the genotypic ratio, that is 1:2:1. So 1 and 2 forms the 3 of the phenotype, and the last one, that is always the recessive form. Like this, he could test all the 7 characters and he found the same results. After this, he could formulate 2 laws, that is law of dominance and the law of segregation. What I have just discussed can be shown here. Tall and dwarf got the tall plants, F1 tall plants, F1 tall plant crossed, and here you find 3 tall and 1 dwarf. This is what we have observed and Mendel found during his experiment. Now, how did he get this? That was the observation which he actually experimented and found. To explain this, how these things are possible? These things are possible by considering this. Let the genotype of the tall plant be capital T, capital T. Capital T, capital T, we are denoting tall plants as capital T and the dwarf, which is recessive, as small letter of the dominant traits. So here dwarf is small t. Since these plants, they are diploid, so the genes will be 2. Capital T is a homozygous condition. Small t is a homozygous condition. So these are homozygous plants or pure plants. What he found that capital T and small t, they will separate during gamete formation. This is actually segregation. So here you can see capital T and capital T, they are separating. Small t and small t, they are separating during gamete formation. This is actually segregation, or this is the principle of segregation. So after this, what happens? That is, along this axis, capital T, along this axis, you can find small t. Now the question is their probable combinations. This is a Punnett square. That is, what are the probable combinations? Capital T may fuse with small t and form capital T small t. Capital T may fuse with this one and form this one. Capital T and this small t, and capital T and this small t. So all the four are capital T small t. That's why I said that the F1 generation are 100% tall. But this tall is quite different from this tall. The difference is this parental tall plant is pure and the F1 generation tall plant is hybrid. Now, in the next step, what he did, that is selfing of the F1 hybrids. Capital T small t cross with capital T small t. Here again, during gamete formation, segregation occurs. Capital T small t separates. Similarly for this plant, capital T small t separates. Now in this checkerboard or the Punnett square, here you can plot the probabilities. Capital T with capital T forms capital T capital T. Capital T with small t here becomes small t capital T small t. Similarly in this box, capital T and small t comes to capital T small t, and in this small t and small t they become small t small t. Now here you find these three are tall and this is dwarf. But so the ratio is 3 is to 1. Phenotypic ratio is 3 is to 1. 3 tall is to 1 dwarf. But if you see the genotype, this is capital T, only 1. Capital T, capital small t. There are two such, and this is 1. So 1 capital T, two numbers of capital T small t, and one number of small t. So the genotype ratio becomes 1 is to 2 is to 1. Punnett square, just now I have discussed. This was developed by Reginald C. Punnett. What is Punnett square? This is a graphical representation to find probability of all possible genotypes. As we have just discussed, that through this we can plot the possible genotypes. We have also discussed in the case of tall plant how the gametes are formed, segregation occurs, and what happens in the random mating. Now, Mendel's proposal based on monohybrid cross. So he formulated two laws first, law of dominance and law of segregation. Now different authors consider in different ways. Some say that Mendel had three laws, some say that there are two laws. Law of dominance is actually, it is the principle on which the inheritance is based on. So there are two laws, law of segregation and law of independent assortment. So different books we find different interpretation. Let us consider that these are the laws. So law of dominance and law of segregation. Law of dominance says that only one of the parental trait will express in the F1 generation, as we have already studied. The other trait will never express in the F1 but will be able to express in the F2 with lesser frequency, as we have just seen. So this law states that when a cross is made between two individuals of a species differing in one pair of contrasting character, the trait which appears in the F1 is dominant trait, while the one which is not expressed is the recessive trait. Law of segregation states that where there are two factors for the character, each factor expresses one trait. Now, on analyzing the F2 phenotypic ratio, Mendel found presence of hybrids in the dominant trait. So the genotype ratio came to be 1 is to 2 is to 1, where 3/4 dominant trait existed in two forms. One is pure form, another is hybrid form. What is law of segregation? This states that the two factors in any generation, here in F1, it is the combination of one factor from the father and the another factor from the mother. So these factors, they always will exist in a paired condition, except in case of gametes. And fusion of the factors, they occur at random, as we have just seen in the monohybrid cross, during mating of gametes, and finally establishes the pair condition. So these factors in the parent, they were in twos, means there were two doses. In the gametes, they become single dose, and again in the zygote, they become double doses. So this separation of factors while gamete formation in any generation is actually segregation. So allele pairs that remained together separates during gamete formation, and the paired condition is restored by random fusion of gametes. This is actual law of segregation. Now we come to another interesting part, that is, we have already understood what is monohybrid cross and what are the different types of characters studied, and got the same results, 3:1 and 1:2:1, phenotypic and genotypic ratios. Now, test cross and back crosses. These two are important tools to identify certain things. Let us see. A test cross. In this experiment, here you can see that this is a flower. This is a flower. This color is violet. Violet color pea plant, or violet color of the flower in pea, is a dominant phenotype, whereas white color of the flower is a recessive phenotype. Here let me tell you that recessive phenotypes always have homozygous genotypes. The recessive trait is always homozygous. So if we consider the allele here, capital W and small w, then the white flower, since it is recessive, will have two alleles, small w, small w. This is the violet flower, which I find here to be a dominant trait. So I know the phenotype of this flower. Do I know the genotype of this flower? No. Why not? The reason being, the dominant trait, the dominant trait here can be homozygous or heterozygous, which we don't know. But if we conduct an experiment, we will be able to know whether it is a dominant or whether it is a dominant homozygous or dominant heterozygous. For this, let us consider, that is, to know the genotype of this plant or this character, this will be crossed with an individual having a recessive genotype. That means this plant, this flower, now has to be crossed with a recessive flower, that is white flower. Now what are the probabilities? There are two probabilities. If the result is 100% violet, then this unknown genotype is homozygous. If this results into, or the cross results into 50-50, means 1:1 ratio, 50% flowers are dominant, means violet color, 50% flowers are white color, then it is understood that the genotype is heterozygous. This can be explained by this cross. Here you see, if this is heterozygous, capital W, small w, they segregate capital W, small w, cross with this white flower having small w. Now, this is now plotted in the checkerboard, and easily we can get that here there is one capital W, so all of them will be violet flower. In this case, small w, and in both the cases it is small w, so these two combines to form white, this and this combines to form white. So here in this we have 50-50, that is 1:1 ratio. So if you just go the reverse way, so in the test cross, if the result is 1:1, then you must consider that this flower is a heterozygous type. If this flower is homozygous, then what happens? Then both the alleles are capital W, capital W. When both the alleles are capital W, let this be, there is segregation here, and small w, they segregate here. Since there is one capital W, and here also in one capital W, so ultimately all are heterozygous. So all flowers are violet, means 100% violet flowers. That means if we go into the reverse way now, let us try to interpret in this way. If the cross results into 100% dominant trait, that is the violet flower now here, then the genotype of this plant or the flower will be homozygous dominant, that is capital W, capital W. So this is how a test cross can be useful in identifying the genotype of this phenotype. Now let us go to back cross. Back crosses means when we cross the F1 plants with genotype of their previous generation, that is genotype of their parents. It is not possible to cross with the parents. So you will now identify plants having similar genotype of the parents and cross the F1 plants. This is called as back cross, means crossing with the back generation genotypes. These crosses are usually done in animal or plant breeding programs. Why? To improve the varieties by making homozygous or pure lines of the desired traits. Now let me remind you that test cross, because in the back cross, here there can be two types of crosses. That is, F1 generation can be crossed with the recessive parental genotype. The F1 can be crossed with the dominant parental genotype. So when we cross, so both of them are back crosses. But when we consider this F1 generation crossing with the recessive parental or recessive genotype, it becomes a test cross. So a test cross is a back cross. But all back crosses need not be test crosses, because if F1 is crossed with the homozygous dominant parent, it is not a test cross, but it is a back cross. So we have completed the concepts on monohybrid crosses. Now let us discuss dihybrid cross. Dihybrid cross means when we are considering the inheritance pattern of two characters at a time. Here, what we do, you can see the diagram here. This is yellow round seed, pea seed, and this is green wrinkled pea seed. So the parents are pure, yellow round and green wrinkled. RR, YY, all capitals, they are dominant traits. So two characters are taken here. One is round shape, that is shape of the seed, and second is color of the seeds. So here we have taken two characters. So study of two characters or inheritance study of two characters at a time is called as dihybrid cross. The two alternate traits are shape is round, shape is wrinkled. Color is yellow, color is green. Through separate monohybrid crosses, we have found that round is dominant over wrinkled and yellow color seed is dominant over green color seed. So now we choose two parents. One is yellow round, that is homozygous, and another is green wrinkled, that is homozygous. From each, now the gametes are formed. So here gametes will be capital R, capital Y. Here small r, small y. You cross them, you get as usual capital R, capital Y, small y. Since there is at least one dominant gene or factor for each trait, so the character expressed will be round and yellow. But this round yellow is a hybrid because you see capital R, small r. This is a heterozygous type. Small capital Y, small y. These two alleles are heterozygous. Now, when we go for the selfing of the F1 generation, capital R, small r, capital Y, small y, they will form now the gametes. Naturally, these gametes formation are probabilities. Capital R may go with, now during gamete formation, this actually happens when gamete formation takes place, then capital R may go with capital Y, here and see. Capital R may go with small y, here you see. Small r may go with capital Y and small r may go with small y. So these are the four different types of gametes which can be formed. So they are plotted. Male gametes this side, female gametes this side, and we find 16 such probabilities. 16 possible combinations. Means 16 possible here, this is the genotypic combinations. Now out of this, when we consider their phenotypes, what we find? 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 are round yellow. 1, 2, 3. These are yellow and wrinkled. These 3 are round but green, and one is green and wrinkled, or 9, 3, 3, 1 ratio, which is the phenotypic ratio of the dihybrid cross. The same thing we have just seen in the previous slide. This is how the cross occurs. 9, 3, 3, 1 is the phenotypic ratio. Now how he got this result? Mendel chose 556 seeds from the F2 plants and found that 315 seeds were yellow round, 101 were yellow wrinkled, 108 were green round, and 32 were green wrinkled. And now making these ratios, we get this is 9, 3, 3 and 1. Now how this ratios actually develops? 9331 ratio can be is actually a combination of two monohybrid crosses. As I have told you that the hybrid cross is a cross where two characters and their inheritance is considered simultaneously. Two characters means there can be two monohybrid crosses. So now let us try to compare the two monohybrid crosses. See, one is round and wrinkled seed. This is the shape of the seed. Round is dominant over wrinkled, so the F1 ratio is 3:1. Similarly, color of the seed, yellow and green. Monohybrid cross gives a ratio 3:1. So these are the results of the two individual or separate monohybrid crosses. When we consider them together, then what happens? Let us try to see. Yellow round. When we consider together yellow and round, these are two dominant traits. 3 into 3 is 9. Yellow wrinkled 3 into 1 is 3. Green and round 1 is to 3. 1 into 3 is 3. And green and wrinkled 1 is to 1 is equal to 1. This is how the 9331 combination appears. Now, out of this 9331 combination, don't you find some new appearance? Yellow round was the parental combination, green wrinkled was the parental combination of the two characters. But after this, we find there are two new combinations. That is yellow wrinkled. First, yellow was with round, green was with wrinkled. But now there is a new combination, yellow and wrinkled, green and round. Green was supposed to be with wrinkled, but now we have green and round. These two new combinations are called as recombinants. Now, after this, Mendel came to the next law, that is law of independent assortment. He said that these two new combinants or new two combinations are possible when yellow movement of the gene by the factor for yellow is not dependent on the movement of the other character. So the two characters Y and R, the genes for the two characters Y and R, they have independence. They can move independent of another, and that is why during this type of movement, this new type of combinations would be made. So yellow, which was initially with round, now can go with wrinkled, because these two genes or the factors, they are independent to move in the gametes. So from this, as we have just discussed, the new combinations of yellow wrinkled and green round, which are not parental combinations and are called as recombinants. And these new recombinants are possible since movement of Y and movement of small y, they are independent of the movement of capital R and small r. So capital Y and capital R, they were together in the parental combination, but now capital Y has gone with small r and capital R has gone with small y, and so new combinations could be possible. But one thing is important, that is independent assortment is possible only when the concerned genes or the factors, they are separated on separate chromosomes. That is, Y and R should be present on separate chromosomes. This is as per the later on knowledge. With this, we come to an end of the dihybrid cross. So you must be able to write the different crosses as we have discussed here. Making this checkerboard or the Punnett Square, this is very, very important. Writing the genotypes and also finding the phenotypes. We will give you some questions based on the discussions we have just made so that your learning becomes permanent. Some questions I am giving you. With the help of a monohybrid cross, explain the law of dominance and law of segregation. When is a test cross performed? How many different types of gametes can be formed from the following genotype? This is a very, very important question. And what are those genotypes which can be formed? So now let me say capital A, small a, capital B, small b. Next is capital Y, capital R, small r. Next is capital Y, capital R, small r. And the next is capital A, capital B, small b, small b. So from here how many types, from here how many types, from here how many types, and from here how many types of gametes can be formed and what are they? One interesting question. This is very much similar to what we have studied in the dihybrid cross. That is, you can read here, in dogs, barking trait is dominant over silent trait, and the erect ear is dominant over drooping ear. What will be the phenotypes when dogs heterozygous for both the traits are crossed? And a dog heterozygous for both the trait is crossed with a silent drooping dog. Now show the cross. Very, very important and interesting question also. If you have understood the various terms, heterozygous, homozygous, and the dihybrid cross, then you can do this easily. The last question is differentiate between segregation and independent assortment. Very, very important questions. So with this, we come to an end of today's discussion. I hope you have understood the main points of today's discussion: monohybrid cross, dihybrid cross, test cross, and back cross. And with this, we end here. We'll be happy to see you in the next topic. That is, till now we have understood Mendelism, but in the next discussion, we'll have a detailed discussion on certain deviations from Mendelism. So, thank you for watching and thanking you for being with me in this session. Have a good day.